Hello, this lecture is about cellular adaptation to oxygen, the work behind Nobel Prize for Medicine in 2019. We'll talk first about the background and the work timeline, how the laureates have reached their final perspective on hypoxia-inducible factors, the factors of interest in our lecture. Then we will present the final outcome in some subsequent diagrams to show up the different various regulatory mechanisms for them, and also the genes to be regulated by hypoxia-inducible factor 1 alpha, and how these functions could affect our understanding to normal physiological processes, and how their alterations in diseases. Moreover, how these factors could be used as promising targets for making useful, effective drugs. Last but not least, we will pass quickly on our laureates' personal and career lives. What were the motives for their works, and how that affected their lives. First, let's discuss the work timeline for reaching the final mechanism, explaining the origin and regulation of our proteins today, HIF proteins. Let's begin with how it's discovered. Uh, they noticed first that people at high altitudes, due to the hypoxia, have higher levels of hemoglobin and RBC's number. Hand in hand with that, they detected the hormone erythropoietin, secreted mainly from kidneys, and was proven to be the culprit behind that much erythropoiesis. Uh, and we know that its gene has the same name, must have nearby regulatory genetic segments, and they recorded that. Then Semenza, one of the Nobel laureates this year, could detect the protein complex that can bind to this genetic regulatory segment and called it by hypoxia-inducible factor. And this is, by the way, misnomenclature, and we will expound on that lately. Semenza could purify that protein, and the purification results show up. It's of two dissimilar subunits, and by that he could know well two things. Number one, what hypoxia-inducible factors structure is, and the second is that they happen to exist with low oxygen pressures. Here are the different subunits sharing in the structure of hypoxia-inducible factor heterodimers. We have the constant DNA able to bind subunit beta, hypoxia-inducible factor beta, and the other subunits, any of them could bind to the beta subunit to make up the hypoxia-inducible factor heterodimer. Two functional domains or sites on the different subunits. One alpha ones, we can see the oxygen-dependent degradation domains ready to catch oxygen atoms available in normoxic conditions, preparing it for degradation. On both alpha and beta subunits, we can see the transactivation domain to cause the hypoxia-inducible factor to be able to bind to different genes and thus performing their different functions. Other parts within the architecture are important too, like the helix loop helix parts there, very important to keep the stability. an ability to function for these proteins. And another left sites are path sites, and they are important for alpha subunits to bind with beta ones. Passing from molecular biology to molecular oncology, von Hippel-Lindau protein was accidentally proved to be germane to our scheme. In cancer cells lacking the protein, they again accidentally found that hypoxia-inducible factors were upregulated, but with providing normoxic conditions to cells with presence of von Hippel-Lindau protein, hypoxia-inducible factor vanished. Moreover, they could build up a statistical association between them. And moreover, they concluded that von Hippel-Lindau plays a role in the process of hypoxia-inducible factor degradation via ubiquitin tagging. So we have hypoxia-inducible factor on one hand, and on the other one we have von Hippel-Lindau protein. But how can hypoxia-inducible factor be mediated to join von Hippel-Lindau for degradation? More specifically, how can oxygen be involved in the process? And here comes the hydroxylases enzymes that take oxygen and give it to proline residues at specific sites. And by that, it allows hypoxia-inducible factor to be ready for von Hippel-Lindau binding and degradation process. And vice versa, with no oxygen, the hypoxia-inducible factor is not able to bind to von Hippel-Lindau protein, and they survive the degradation. So oxygen, as we see, shifts the balance, and it's considered in the first steps for HIF to be degraded. This slide is very important because it summarizes up the whole work. Number one, the heterodimer HIF bind to the hypoxia response element that happens to exist in the neighborhood of the hypoxia-inducible factor regulated genes. Number two, the von Hippel-Lindau protein and its role to help HIF1 alpha degradation via the ubiquitination process. Number 3 and 4, how oxygen binds to its sites on hypoxia-inducible factor 1 alpha, and how that binding prepares these subunits to catch von Hippel-Lindau and to get degraded eventually. And all of these steps happen in normoxic conditions. However, the number one process takes place with high amount of HIF under, of course, hypoxic ones. Let's present another angle for the big picture of HIF regulation. We mentioned the oxygen-dependent degradation, but is that the only factor that has dominance over hypoxia-inducible factor? Studies reported no. The answer is no. On the enzyme for hydroxylation of proline, iron binding site does exist too, and of course it doesn't bind to oxygen but to iron. So in the absence of iron, as in cases of bacterial infections that drain iron and its stores, we can record no function for these hydroxylases and vice versa in the existence of iron. So that's another regulatory factor for proline hydroxylases. Other hydroxylases exist but not for proline. They can cause the hydroxylation to asparagine and that mediates the proteasomal degradation directly, even without the need for von Hippel-Lindau particles. Away from oxygen, hydroxyl groups, and hydroxylases, we could identify some different mechanisms for HIF regulation. And that's what takes place mostly within cases of bacterial infections, with activation of toll-like receptors and T-cell receptors. Nuclear factor kappa beta and mTOR pathways are stimulated respectively. So HIF upregulation from these cells deploy and help its survival under infectious and inflammatory conditions. Before we shift gears and discuss the various genes affected by HIF, this year we all witnessed the Amazon forest fires, and many people were in doubt if these fires would affect the climate change of the nearby land or the nearby ecosystem. But here it is. The Amazon is considered the lung of the earth, and its uprooting will not only affect the climate through evaporating gases and accumulating CO2, but also affect the net oxygen pressure all over the globe. And while we are diving through the next slides, you will understand in depth how that could affect our health. Maybe you would be surprised that oxygen pressure, when it slashes, it will cause people to get cancers. It will affect the different embryogenesis phases and may lead to different congenital malformation. So it's very interesting how changes in Earth atmosphere could affect health deeply like that. I guess it's so exciting to understand it. That's what we are trying to do next. So the different genes affected by the different HIF agents, I think we have discussed erythropoietin enough earlier. We will focus more on the metabolic reprogramming, different mechanisms, and the other mechanisms playing an important role in cancer and in inflammation, like angiogenesis, cytokines, and setting up metastatic niches for primary cancers. I think we have discussed erythropoietin enough earlier. Okay. So we will go through the other factors and the mechanisms next. Okay, metabolic reprogramming. Instead of cellular dependence on aerobic metabolism and tricarboxylic acid cycle, HIF switches the dependence and keep up the glycolytic reaction cascade. In detail, HIF stimulates the genetic expression of lactate dehydrogenase and pyruvate dehydrogenase kinase 1, which in turn will inhibit the enzymatic activity of pyruvate dehydrogenase via phosphorylation. So the net result is switching from the acetyl-CoA production and tricarboxylic acid cycle to lactate formation and anaerobic glycolysis. How can that be good or optimal for cellular survival, especially under hypoxic conditions? Because this switch protects against formation of premature reactive oxygen species on complexes 1 or 3 in electron transport chain that could happen if tricarboxylic acid cycle and electron transport chain persisted with hypoxia. In other words, there wouldn't be enough oxygen to complete the chain till water production. Moreover, hypoxia-inducible factor upregulates BNB3 and BNB3L proteins thought they play a role in mitochondrial autophagy. When they accumulate, they competitively bind to BCL2 protein, thus freeing peclin 1, which participates in autophagy process. So, in brief, metabolic reprogramming by HIF goes via two mechanisms: the glycolytic switch and the mitochondrial autophagy facilitation. Both are optimal for the cellular survival. Another accessory mechanism for HIF to do that is by microRNA 210. The net result is the same: inhibition of tricarboxylic acid cycle and electron transport chain. But in this scenario, it makes that by microRNA 210 ability to block iron sulfur cluster assembly protein, and this in turn breaks down the iron dependent centers at aconitase and electron transport chain complexes. These centers are very important for these enzymes to function, also for the redox actions that occur by them to facilitate electron transport. This blocking turns these molecular mechanisms upside down. The diagram, I think it's illustrating itself. HIF1 stimulates COX-4-2 and suppresses COX-4-1, but indirectly via LON protein, which is a kind of degrading protease enzyme, and vice versa in hypoxia. Therefore, we can say hypoxia converts the abundance of COX-4-1 to that of COX-4-2. Why does that happen? All what we have in our hands until now is that under hypoxic conditions, if COX-4-1 wasn't replaced by COX-4-2, reactive oxygen species would rise up and the cellular death is inevitable. We can get a glimpse of more detailed explanation when we studied the homologs of COX in yeasts. With more alpha units, which are analogs for 1 units, the turnover increases. This wouldn't be optimal for hypoxic conditions in which we need more and more of COX. So COX-4-2 would be vital to keep up the levels of COX needed to transfer the electrons under the hypoxic conditions, and not to stacking a lot of reactive oxygen species trash. So the hypoxia-inducible factor 1 alpha are to keep the reactive oxygen species within bearable amounts for the cell to detoxify and to get rid of them. We could see the different mechanisms for that. The mechanisms briefed here, and in some experiments where they knock out hypoxia-inducible factor 1 alpha in mouse embryofibroblasts, they noticed reactive oxygen species accumulation, and they could reverse that by forced expression of pyruvate dehydrogenase kinase 1. So the mechanisms, as we see, are evident and could be of therapeutic value as well. Angiogenesis, different angiogenic and growth factors can be upregulated with HIF1 alpha. These factors themselves, and by functioning as chemotactic factors for bone marrow derived angiogenic cells, can perform the task. And angiogenesis can play a role both in health and disease. Without it during embryogenesis, we couldn't have blood vessels at all, and in wound healing it can play the major role. But in diseases like if it's over regulated in embryogenesis, we can end up with congenital malformations. And of course, in cancers, it's one of the main cancer hallmarks. In experiments where they injected adenoviruses called EDCA5 carrying the HIF factors in wounded mice and followed that by systemic injection to bone marrow derived angiogenic cells, they could get the wound healing. Okay, uh, and that wouldn't happen if we changed the order of these two steps. So there is ordered collaboration between angiogenic cells, bone marrow derived angiogenic cells, and angiogenic factors upregulated by HIF proteins. Uh, by oxygen independent mechanisms, as we have shown earlier, HIF1 alpha is increased in infections and in inflammation. Again, as we mentioned, it builds up different angiogenic growth factors and metabolic reprogramming that help immune cells survive and perform their protective functions. Moreover, we can see specific genes for immune cells can be upregulated by HIF1 alpha, nitric oxide synthase, for instance, and proteins for phagocytic actions. In cellular growth and survival, the pathways of HIF1 modulated signaling in cell proliferation and anti-apoptosis under hypoxia. Uh, Hypoxia upregulates the growth factors and their receptors to trigger signals through RAS, RAF, MYC, ERK cascades, and that results in number one, induction of target genes to inhibit apoptosis and or promote cellular proliferation and survival, and number two, phosphorylation and inactivation of BIM, BAX, PAD, and caspase 3 to block apoptosis. About the cellular metastasis, different mechanisms by HIF to stimulate different cancer hallmarks, as we can see, but one of the most interesting mechanisms is setting up the metastatic niche. Uh, this is number 5. But um, and that was studied clearly in breast cancer. They concluded that um, hypoxia-inducible factor through LOX enzyme, lysyl oxidase enzymes, can remodel collagen in the potentially metastatic sites, and that makes the first steps for the distant sites to be deployed with cancer cells. So from the previous slides, we can conclude the various physiological and pathological impacts of hypoxia-inducible factors. The impact on metabolism and exercise through metabolic reprogramming, on embryonic development through vasculogenesis, and on wound healing through angiogenesis and cytokines production. The effects of metabolic reprogramming and chemotactic cytokines on immune cell survival and response. We could understand the response to RBC's inability to carry enough oxygen through erythropoietin and upregulation. Also, the collaterals formation when HIF produced for relatively short time in myocardial infarctions and strokes. For more details, you can browse the references we armed with the presentation below. Last but not least, the Nobel Laureates. The Nobel Prize for Medicine this year is awarded to William Killen, Sir Peter Ratcliffe, and Greg Semenza on the vastly discussed topic. All of them are medical doctors. Dr. Semenza is a former pediatrician and currently affiliated at Johns Hopkins University. Sir Peter is a nephrologist and a professor at Oxford. Dr. William Cullen from Dana-Farber Cancer Institute, and I think it was such a touching photo, the one he shared just after the prize announcement, a selfie for him and a portrait for his wife, the breast cancer surgeon who died around 3 years ago. So three great scientists and doctors from three highly ranked universities. And as we say here, they only lack two soldiers to declare a war. So hope you enjoyed. Thank you.